Good morning to everyone, and good afternoon for the people located in Europe. And it's a great honor to participate in this plenary lecture. And in this sense, I want to introduce the first speaker, Ines Borthagaray. I shared a few slides in order to introduce Ana. Okay. Ines Borthagaray comes from Uruguay. She born at the end of the decades of the last century. I am not allowed to give more details about this. In 2002, she obtained her bachelor degree. In 2006, the master degree in biological science, ecology option in the same University of Uruguay, Universidad de la República. And in 2011, she obtained the PhD also in biological science, ecology option in Pontificia Universidad Católica de Chile, located in Santiago de Chile. Since 2014, Ana has been a professor in a regional center of the Universidad de la República, that in this case is located in the East region, Maldonado, Uruguay. And she's a member and integrate the Department of Ecology and Environmental Management. The main research topics of Ana is related to the effect of the spatial structure of the landscape on the pattern of biodiversity and ecosystem functioning, and also the role of the microscale variability as a key factor or drivers that determine the stability at macro scale. In this case, also paying attention to the most global and environmental change. Finally, I want to introduce you a few examples of papers contribution of Ana. I select a few of them, most directly related to the plenary lecture. And in this sense, welcome, Ana, and I hope you enjoy this session.
Thank you, Nestor. Thank you very much. Hello, everybody.
Thiago, can you, yes, please. Thank you.
Thanks for this invitation. It is a pleasure for me to be here. This talk is about the role and importance of considering the metacommunity network structure to advance on the mechanisms that determine biodiversity patterns. In the bottom right corner is a slide number in case there are doubts or questions later. So, ecological theory has focused primarily on local determinants of species coexistence as a main approach to explaining biodiversity patterns. Although the role of the species pool, local community connection, was early identified as a determinant of diversity, the process operating within communities have dominated the attention of ecologists, specifically local process associated with biological interactions as competition and interpretation, and environmental conditions have been considered the main determinants of community structure. This vision of communities as essentially isolated entities structured by local process was replaced by a vision wherein the connection of the local communities to a regional pool of species can be an equally or more important determinant of its structure. In fact, early proposed biogeography highlight the role of connectivity on species assemblages, wherein the colonization rate or dispersal is a function of the distance to the continent or source area. So more recently, the abstract concept of a regional pool of species has been replaced by a vision where each community receives a flow of individuals from the neighbors communities. So a network of communities, and that is what we call a metacommunity, a set of local communities connected by the flow of individuals among them. Thus, an important change in contemporary ecology has been the recognition that approaches focused primarily on local determinants cannot account for biodiversity patterns. In this framework, the flow of individuals in the landscape is a main determinant of community structure. In this context, considering an explicit metacommunity network, immigration is frequently composed by neighboring communities, and each community has the potential to act as a source of immigrants for the other communities. Thus, the flow of individuals and its magnitude is affected by species dispersal abilities involved in the metacommunity assemblage, associated with body size and dispersal mode, and also by the landscape structure, that is the spatial arrangement of communities in the landscape. So the flow of individuals among communities and its dependence on the landscape structure have a key role in biodiversity patterns and ecosystem functioning, and this is supported by several studies that demonstrate the importance of considering metacommunity network structures, not only on species richness, but also on functional diversity, spatial and temporal turnover, trophic structure, body size structure, and ecosystem functioning. However, the assumption of relatively homogeneity in the flow of individuals and the poor representation of landscapes configuration have limited the understanding of more realistic natural systems. So, in this sense, two main challenges are that first, on one hand, we need better representations of landscape configuration in order to identify metacommunity networks that allow a more realistic quantification of the flow of individuals that a community receives. This is a more methodological challenge with, of course, theoretical and empirical implications. On the other hand, several models have been significantly contributed to understand the role of the flow of individuals on diversity patterns using mainly theoretical networks or implicit spatial networks. So we need to advance on models that connect theory with empirical patterns. So herein today, I would like to discuss three issues on the interplay between metacommunity networks and biodiversity patterns. First, about quantification of metacommunity networks, then the role of isolation on community structure, and finally, the potential of lottery models for the theoretical and empirical evaluation of metacommunity mechanisms. So to start, one important point is to achieve better representations of more realistic landscape structure, and in this line, the use of graph theory for metacommunity studies has been emphatically recommended in recent years. However, the methods by which networks may be empirically determined are not always so obvious. So here I would like to also present alternative quantifications of empirical metacommunities. So graph theory has long been used as a framework for the analysis of ecological networks, considered as food webs, mutualistic networks, and increasingly in landscape ecology. In this line, graph theory has positioned it as a main, spatially explicit approach to metacommunity and landscape ecology. In this context, a metacommunity can be described by a network or graph defined by a set of nodes connected by links. Typically, nodes correspond to communities, and connections refer to some kind of structural or functional relationship among them. For example, the flow of individuals. Once defining the graph theory provides a wide set of tools for quantifying the structure at the level of the whole network and at the level of individual communities. That the metacommunity structure or configuration is defined by the spatial arrangement of communities in the landscape, wherein the spatial location or the position of communities in the landscape determine the degree of connectivity in relation to other communities. And this determine the flow of individuals to which communities are subject. So here are 3 examples of landscapes with different spatial structures. The first is a riverscape represented by a hierarchical structure, typically directed. The second corresponds to a seascape of rocky shores, represented by a chain structure. And the third is a pondscape, represented by an unknown a priori structure. Metacommunity studies need an explicit representation of these networks, and graph theory provides tools for representing these explicit representations of networks and quantifying their structure at the level of the whole network and at the level of individual communities. However, these representations are possible when we know the configuration of the empirical metacommunity. For example, a natural network configuration, as could be riverscapes with dendritic networks, or similarly using molecular or genetic approaches, it is possible to establish the connectivity pattern between communities. In that cases, we can define the empirical metacommunity network. However, in most cases, metacommunity configuration is unknown, or the level of sampling efforts required cannot be achieved. So in that case, how can we define the empirical network configuration? So we have in one extreme the pond's location in the landscape, and in the other extreme, a fully connected network wherein all communities are connected to all communities. Probably the empirical network configuration is in one place of this gradient of connections. To use it, approximations for estimating spatial networks are the minimum spanning tree and the percolation network. And here I would like to present a third approach that is called taxon contingent metacommunity networks. So the minimum spanning tree is the graph, the network that connects every local community with the shortest path length, that is with a minimum number of links to ensure that all patches are connected to a single graph. In a metacommunity context, this network represents the backbone of the network, maximizing the flow of individuals with different dispersal abilities among communities. The other alternative, the percolation network, is defined as a graph in which communities are connected when the distance between them is less than a threshold distance, that is called the percolation point, and is defined as the minimum distance at which all patches are connected in a single component in that network. In biological terms, this distance implies a qualitative change in the flow of individuals through the landscape. And this is congruent with the fact that the ability of organisms to disperse among communities typically fits a pattern of abrupt transitions from movements on local scales, that is, organisms perceive fragmented landscapes, to movements that allow potential access to all parts of the landscape beyond the critical threshold distance. So, although the minimum spanning tree and the percolation distance network provide robust estimations of metacommunity networks, they are not built from biological data. So they may differ significantly from the landscape perceived by the organisms or fully represent the process shaping local community structure. So here we propose a third approach called taxon contingent metacommunity network. The idea behind this is that organisms in the same landscape perceive the different degree of landscape fragmentation. So dispersal ability associated with body size or other biological attributes determine the linkage distance to connect local communities. So for individuals with short dispersal ability, the metacommunity network will be essentially fragmented, while for individuals with high dispersal ability, the metacommunity network would be perceived as more connected. Consequently, the alternative metacommunity network structure will reflect the different landscape perceptions for different taxa. And among all the possible networks, the one that best represents the dispersal capacity of the taxa should achieve a better explanation of the metacommunity structure. So briefly, this approach consists of 4 steps. First, constructing a set of metacommunity networks along a gradient of linkage distances to connect to communities. That is, then the estimation of community isolation and richness for each metacommunity network. Then determining the association between the isolation and the community attribute, richness in this case, for each community of each the metacommunity networks. So in this case, we are going to have a correlation value for each metacommunity network at each linkage distance. And finally, identifying the linkage distance that maximize isolation community attribute. That is, the underlying idea behind this is that a community attribute, community isolation association will have an extreme value at a linkage distance congruent with the distance affecting organisms flow. So for each biological group, we have a metacommunity network structure that best explains the local diversity. In other words, a metacommunity network based on maximization of coherence with a community attribute. So until now, we discuss about the importance of community networks to advance on biodiversity patterns. And since the flow of individuals is a main determinant of biodiversity patterns, considering explicit metacommunity networks seems to be more important than previously thought. And in consequence, the estimation of confident explicit metacommunity networks in empirical studies is of most importance. Continuing with this talk, I would like to focus now on the role of isolation on community structure, metacommunity structure. So, what is the effect of community isolation on local community structure? We know that more central communities are expecting to receive more immigrants, and this promotes an increase in population densities, colonization of new species, and the persistence of sink populations. Of course, this is the general picture. This effect could be differentially impact organisms of different body size, depending on dispersal type and trophic rank. In addition, community isolation has been frequently estimated as a categorical variable, that is with few, for example, few levels of dispersal, that is low, intermediate, and high dispersal, connected versus isolated patches, or as a distance to a source of propagus, for example, distance to the outlet in a riverscape, to the continuous forest, average distance to the closed patches. However, as we see before, in the last years, the importance of considering the spatially explicit structure of the metacommunity network to advance on biodiversity patterns has led to rethinking the concept of community isolation. So the metacommunity network structure defined by the spatial arrangement of communities in the landscape promotes an isolation centrality gradient, which is used as a proxy of the flow of individuals that a community receives. Along this gradient of isolation centrality, a continuous measure of community connectivity is available with network centrality metrics. In this context, communities with high values of an isolation centrality metric are located in central positions in the landscape, playing a critical role in the dispersal pathways across the network. Despite the recognizing importance of this isolation centrality gradient, from the sampling design to the analysis of patterns, few studies have used a continuous representation of the isolation centrality gradient of local communities. Thus, the network centrality measures quantify the isolation of a local community, potentially reflecting the individual flow among communities and through the whole metacommunity. In a metacommunity context, the 4 major centrality metrics used are degree centrality, agent vector centrality, closeness centrality, and between the centrality are the 4 presented in this graph. Larger values of centrality indicate the lower levels of community isolation, and alternative centrality measures complement each other in the representation of different components of community isolation. So let's go to define these metrics. First, the degree centrality is the number of direct connections between a community and its neighbors. That is, direct links between patches. The term connectivity of a node has also been used to refer to this centrality metric. Then agent vector centrality ranks communities not only by the number of direct connections, but also by the number of connections that the neighbors have. In this sense, it may be seen as an extended degree centrality since it is proportional to the sum of the degree centralities of the community neighbors. And the two other definitions, closeness centrality and betweenness centralities, are both based on the length of the shortest path between communities. Closeness centrality is defined as the reciprocal of the average length of the shortest path between the reference community and all other communities in the network. This metric provides a representation of how close or how far a community is from the remaining communities in the metacommunity, and betweenness centrality describes how often a community acts as a mediator on the shortest path between two other communities. This metric identifies key connectors, that is, stepping stones of individual flow to the rest of the metacommunity. So these different centrality metrics emphasize different aspects relating to the spatial scope considered. Degree centrality highlights the potential flow of individuals through the local neighborhood. Agent vector centrality expands the potential flow of individuals to a larger neighborhood. Closeness centrality is a measure of how long it takes an organism to move sequentially from one patch to all other patches in the metacommunity. It shows how far or how far a community is to all the other communities in the network. And betweenness centrality emphasizes the bridging role of the patches in maintaining metacommunity net connectivity beyond the low scale. So, what is the evidence? What is the evidence of the role of isolation as a determinant of community structure? Here, I would like to present some examples about this, but also pay attention to the different metacommunity networks used, and also the different centrality metrics selected in each work. So this is the first one. This is a theoretical study. It's a beautiful and clear study from Economo and Kitt, published in 2010, where they show how node position, how community position predicts variation in alpha diversity across a metacommunity. They represent the metacommunity as a dendritic network of local communities and use network centrality measures to quantify the isolation of a local community. These plots correspond to the spatial distribution of the degree and closeness centrality through the network. Blue colors correspond to low centrality values, while red colors correspond to high centrality values. Then, using a spatially explicit neutral model, they predict alpha diversity for each community. These four plots correspond to the spatial distribution of the expected diversity, considering a gradient of migration. So, what they found? First, they show that diversity patterns at low migration is similar to the pattern determined by degree centrality. Remember that degree centrality was highlight the flow of individuals at the local neighborhood. And in this scenario, diversity is a function of local connectivity, and also look that the central and peripheral communities are similar in terms of diversity. In addition, they show that diversity patterns at intermediate migration is similar to the pattern determined by closeness centrality. Remember, closeness centrality shows how far a community is to all the communities in the network. So in this context, diversity is a function of connectivity at a larger scale, and also the most diverse nodes are the most central nodes. Additionally, they show the variation on diversity, not only the absolute values of diversity as before, along a migration gradients. They found that at extreme of low and high migration, alpha diversity is nearly constant across the metacommunity network. And it is because each node becomes an independent community when migration is low, or the metacommunity is effectively panmitic at high migrations, that is a big community. So in this case, the position, the isolation or centrality of the communities in the network doesn't matter. At intermediate migration values, the highest variation in alpha diversity is observed. In this context is when the position of communities determines the variation in diversity. So this study emphasizes the importance of metacommunity network and the role of isolation on diversity patterns, and also the use of alternative centrality metrics to detect these patterns. So, but this is a theoretical example using theoretical networks. But what happened with the role of isolation in empirical metacommunity networks? So here I would like to present 3 examples of the role of community isolation on different empirical metacommunities, but also considering the role of local environmental conditions as a determinant of community assemblages. The first example corresponds to a study in Uruguay. A pondscape, we analyze a metacommunity of 22 temporary ponds embedded in a matrix of grassland. The pool of species is composed by microinvertebrates, plants, and fishes, conforming 25 groups classified at the level of order. So here we first identify a metacommunity network structure for each of the 25 taxa. Then we characterize the ponds by local conditions, with area, heterogeneity, productivity, depth, and also by isolation centrality metrics using the metrics mentioned before, that is degree as the number of neighbors, closeness as the average distance to all the ponds, betweenness as the number of times a pond is a bridge to connect other ponds. And also we estimate the local diversity for the 25 taxa groups. With this, we evaluate the effect of local conditions and of the metacommunity or isolation centrality gradient on local richness. So what we found first, first, the degree centrality was the metric that had a good performance for the isolation estimation in all biological groups. In addition, we found a significant sigmoidal relationship between the probability of a metacommunity effect on local diversity and body sizes. And that is, big sizes would be more affected by what could happen at the metacommunity scale. So here we have that metacommunity effect depends on body size. Also, among the taxa with significant metacommunity effects, we found a negative effect of community centrality on local diversity associated with intermediate to high abundances, while positive values were observed in groups with low abundances. That is, the sign of the effect of centrality on local richness would be associated with the abundance of the group. So we found a metacommunity effect on species richness associated with mean body size and the abundance of each group. The second example that I'm going to present you is correspond to a riverscape in Uruguay. We represent the dendritic structure of this basin riverscape in a directed graph, quantifying the isolation centrality of each river section. Then 58 fish communities, the one highlighted in sky blue in the graph, were sampled following a stratified sampling design that properly represent the isolation, centrality, connectivity gradient through the riverscape. This is what we that is what I show in this plot, in this histogram. We have the congruence between the isolation centrality of the sample communities and the isolation centrality of the nodes in the graph. In each community, abundance, biomass richness, and functional diversity of species were estimated, and also environmental conditions were determined for each community. So with this, we evaluate the effect of environmental conditions and the isolation centrality gradient on fish community structure. In this case, we use structural equation models to analyze the data. Note that out closeness was the metric of isolation used in this case. And we found a direct effect of the isolation centrality gradient on total community abundance and also on 3 environmental conditions. And through these variables, we detect an indirect effect of isolation centrality gradient on our components of local community structure as biomass, species richness, and functional evenness. So here we also found empirical evidence of direct and indirect effects of isolation on community structure, on fish community structure. The last example corresponds to a landscape in the Atacama Desert, north of Chile. This is a metacommunity of 31 vegetation patches in a matrix of sand. Each community is composed by, the regional pool is composed by 55 species, mostly arthropods, 1 species of lizard, and 1 species of gecko. With this, we evaluate the effect of isolation centrality gradient and local conditions on local body size structure. And we defined, in this case, the metacommunity network with a minimum spanning tree and estimate the isolation centrality gradients with the metrics previously presented. And in each community, we determine local conditions and the body size structure. In this case, specifically, we identified the body sizes at which an overrepresentation or an underrepresentation of individuals occurs in each community. So first, we found in this case that between the centrality was the metric that showed an association with body size structure. We analyzed the data, separating the herbivorous assemblage and carnivorous assemblage, and we found that for the herbivorous size structure, the internal attributes of communities better explain the body size structure. However, the carnivorous body size was more sensitive to the metacommunity structure. Notably, we found that in more connected patches, that is, communities with high values of between the centrality, extreme values of body size, that is, small animals or larger animals, are overrepresented, what we call aggregations. Thus, we showed in this study that empirical evidence of a relationship between metacommunity structure and body size distributions for the carnivorous assemblage. To finish these sequences of evidence of the role of isolation on local structure, I would like to mention this study from Herein. Herein, using a size-dependent model for food chain length in communities subject to migration, show that populations with higher influx of migrants are able to persist at higher trophic positions. This model is simulated on random, dendritic, and real aquatic metacommunities. These plots here show the relationship between trophic position and community isolation, quantified by degree and closeness centrality. And we found that more central patches contain larger populations and were less constrained in trophic position. And these results indicate that community location, the community position in the landscape, may determine local food web structure. So what I try to convince you is that isolation becomes a cornerstone in the metacommunity theory to advance on community structure. I present some examples about the role of isolation on richness, abundance, body size structure, and trophic structure. Also, network centrality metrics are good predictors of the isolation centrality gradients, and important different metrics should be considered depending on the metacommunity network and the species involved. As you could note in the examples presented before, the selected metrics was different in all cases. Finally, I would like to mention that community position in the metacommunity network not only affects the patterns of diversity, as we saw before, but also affect the mechanisms that determine these patterns. So isolation affects the relative contribution of the mechanisms proposed as determinants of the organization of species assembly in metacommunities. Okay. So to finish this talk, I would like to present the potential of lottery models combining with empirical data to achieve predictors more adjusted to real systems, and thus better understand the mechanisms and the role of the metacommunity structure and the role of isolation on biodiversity patterns. So, lottery models simulated on spatially explicit networks have been particularly suitable to advance on the interplay between landscape structure and biodiversity patterns. Also, lottery models are able to capture mechanisms operating at the metacommunity level, not requiring the modeling of every population dynamics, which has been a limitation to advance on theoretical analysis of which community assembly mechanisms. Finally, also, while lottery models were incorporated as a chief tool in the theoretical ecology, their use on empirical studies have been more limited. So, to end this talk, I would like to present a work that is still in progress with Matthias Arim, putting the focus on the methodological approach, rather than the details of the results. So here, in using a lottery-based simulation model, the idea is to quantitatively evaluate the support for alternative hypotheses about the effect of fish dispersal patterns on alpha and beta diversity of a riverine metacommunity, the one that I showed before. So when we think about dispersal, multiple non-exclusive dispersal scenarios are plausible in rivers with different consequences on community assemblages. This example is in a riverscape, but of course we could think in other landscapes. So suppose this is the section of a riverscape and the main flow runs in this direction. The null hypothesis is that communities are isolated. Migration is not able among communities. Also, the dispersal from a regional pool could be independent of the dendritic network. So in this case, the position of metacommunities in the network doesn't matter. Then also the dispersal the dendritic topology may limit the individual movement constraining dispersal. That is. Additionally, upstream and downstream dispersal may be asymmetric. Furthermore, the river outlets typically represent an important source of colonizers upstream, particularly when it is connected to a larger drainage basin, as it is the case. So the distance to the river outlet is in this context a main determinant of incoming dispersal and new species recruitment. Finally, fragmentation by dams and habitat degradation by water pollution interrupt the dispersal process in rivers, so dams efficiency affects dispersal. And finally, we propose that local density increase with community centrality. The idea here is that when we run the model, all communities could have the same number of individuals or could have a density proportional to its degree of isolation in a network. Thus, while dispersal is recognized as a main determinant of river diversity, there are several plausible and non-exclusive hypotheses about the proximal mechanisms through which the interaction between landscape structure and species dispersal-based mechanisms shaped biodiversity patterns. So we developed a lottery-based simulation model and a likelihood framework for assigning support to these competing hypotheses about the role of dispersal mechanisms and river fragmentation by dams of fish alpha and beta biodiversity. Briefly, the basic lottery model is as follows. This is the graph, a section of the riverscape. Each node is inhabited by a community of 500 of individuals, 50 species with 10 individuals each one. And in each iteration, 50 individuals are replaced, are randomly removed from a focal community, and an individual removed is replaced with another from a random sample of the same community with a probability of 1 minus or from the connected communities with a probability of and this is repeated a number of iterations, 2,000, 3,000 of iterations. The output of these runnings are species per community matrix. So the alternative hypotheses are translated, changing the parameters of migration in the lottery model, determining a species by community matrix. So we run the model considering all combinations of hypotheses, and in each case, different ranges of migration values are considered. For example, in the first hypothesis, for the parameter migration pool, 4 values from 0 to 0.5 are considered. Then the expected and observed alpha diversity are estimated. And with a likelihood approach and weight of evidence in support of each hypothesis, the model, the best model is selected. So the weight of evidence of the alternative model found that migration is strongly constrained by river network structure. Moreover, the main direction of river flow determines a larger downstream than upstream dispersal effect. In addition, a large-scale effect of migration from the outlet was detected, and a dam effect shaping diversity at the landscape level also detected. Furthermore, this is the relationships between alpha between the alpha observed and expected and beta observed and expected with the results of the models. And we found that the selected model was able to account more than 50% of the variation in diversity. So, over the details of this particular analysis, herein I would like to emphasize the potential of this kind of approach to advance on the empirical analysis of the community structure and species dispersal-based mechanisms shaping biodiversity patterns. So I have tried to present 3 big issues to advance in understanding of the mechanisms that determine the patterns of diversity, emphasizing the importance to consider a landscape scale, the role of isolation communities, and the use of approaches that are able to connect theory with empirical data in real systems. So thank you very much for your attention. That is.
Thank you very much, Anna.
Thank
Beautiful
you.
lecture. We have a lot of questions. The first one from Stephen de Klerk, first mentioned, great talk. In addition to geographic distance, isolation may also be determined by other factors such as barrier, directionality, or corridors. You consider this also important? If so, how will you incorporate this in your network approach?
Okay, thank you very much, everybody, for listening. Thanks for this question. Yes, of course, you can we can incorporate it in several ways. For example, when we direct the graph, the network, and estimate isolation with the centrality metrics that I present, we can also first we can direct the centrality, that is, we can estimate out degree, in degree, out closeness, in closeness, and in this way you are considering the direction. But also, for example, the barriers, when we define the matrix that represent that network, you are considering the barriers, because maybe two communities are very near. However, maybe you have a mountain in the middle, so the matrix have to say that you are not the distance is not so short. You have to move more, for example. You can weight the centrality metrics, for example, by distance or by other, by the geographic distance or by other distance metric. So, thanks.
We have another question from Nicolas Vidal. Hi, Anna, thanks for this great talk. My question is there any way to differentiate the dispersion in space, for the dispersion in time? Particularly the above, very important for zooplankton community in vernal pools or temporary ponds. By the way, in the example of pond is a slide 42, were not included. Maybe this can affect the relation with the body size.
That study is empirical, so we have the data for space. The time, yes, of course, could affect the pattern, and for example, could be incorporated in a model. Then we have a prediction, and we can contrast with the empirical data. That is, I think.
From Pedro Perez. Hi, Pedro, nice to see you again. Thanks for this great talk, Anna. There are important considerations for many future developments. We have used a spatial graph based on maximizing distributional pattern similar to the way that you have approached. We often find that very different networks can lead to similar biodiversity production. For example, the likelihood function is not unimodal. In this meta community, what are your thoughts about the dispersal dynamic that took place? One possibility is that the same meta community are made of different subnetworks. In other words, the dispersal dynamics is not spatially stationary. Do you have any additional reflection about this? How will you also consider that in your lottery model?
Okay, thanks, Pedro. Big question. First, I would like to say that when we run for example, the lottery model, we consider for the migration parameters in the different hypotheses that I showed, different values, and it is true that we have maybe the same biodiversity pattern with different combinations of parameters, of values of the migration parameters. So I have to say that it is a study that is in progress, and we are working in this. But thank you very much for this for this question. And let me see.
And let's include an additional reflection about the Pedro question that is related to how to wave in an objective way. Steve, I suppose that want to ask how can ponderate or weigh these possibilities.
Sorry, this is the following question of Steven.
Yeah.
Okay. Okay. Sorry. in an objective way, but I don't understand what do you mean. Weight, for example, you said in the other. Weighting the links of the network by a distance, for example, kilometers or degree of similarity among communities. I don't know. I think this is an objective way, or maybe a null model could determine the significance of the links. But a priori, I think you define the links in the graph in an objective way. No?
Thanks, Anna. We have another question from Smith Jansen, sorry for the pronunciation, from Germany. Great talk, thanks. Could you comment on other factors of distance and isolation like land use and agriculture or urbanization?
I think, thanks for this question. I think it is maybe in the line that say Steven, that urban or agricultural land use, we can wait. Maybe it is more difficult to a species or an individual to move between two communities that have a city, for example, or an agricultural land in the middle.
Anna, if it include an explanation about the previous question. I mean, a barrier as a mountain, do you express this as a geographic distance? Is at the end of the question and answer.
Okay. I think, for example, I think it depends on the species that are involved. So, because maybe a species that can fly, a mountain doesn't matter. But if it is a species that have to walk through the mountain, maybe you can measure the real distance, no, not the Euclidean distance, the topological distance between the communities.
Anna, we have another question for Luc Demester. Thanks for the great talk, Anna. Probably a super naive question. You emphasize network on spatial coordinates and how useful they are. Would it also be possible to construct network of trait connectedness or taxonomic connectedness and related them to each other or to the spatial network?
Yes, I think yes. For example, we have a study that we determine the co-occurrence of species in the Atacama Desert, in the system that I showed. And the network is a matrix of species by species, connected by significant links of positive co-occurrence. And then these results were connected with the spatial metacommunity network. Yes, I think the link in a network, you define whatever you want. The link means a functional, structural, a trait, or taxonomic distance or relationship between two nodes that could be species communities, or whatever.
The final question, sorry. Antonio Jimenez, hi, thanks for the talk. You say there is a relationship between metacommunity structure and body size distribution for the carnivorous assemblage. Have you compared this metric with the trophic state of the water bodies or other traits to know which approach are more informative or the difference between them?
I don't know if I understood the question. That study was in the desert. I don't know if you means that with attributes of the patches, in this case vegetation patches. I don't know if this is the question. Yes, we consider the local conditions in the to determine the body size structure of carnivores and herbivores, and only for herbivores the local conditions were significant. I don't know if I answer this question, because I didn't understand very well. Maybe Antonio, you can.
I have a question related to Antonio, that
Yes.
about the results of the vernal pools in the case of the pondscape. That I'm talking about the paper about the influence on the trophic web structure. That's
Okay.
very, very nice results. But I'm thinking the role of the size of the temporary ponds, because I remember in the case of fish, in this case, the occurrence of the fish depends on the size of the vernal pool. Also playing a key role beside the connectedness?
Yes, I think that study, I think, I don't remember exactly, but I think the area of the pools, the ponds were not considered, because if you remember, there are two theoretical networks and the empirical metacommunity. Okay. But of course, the area affect, but I think that also the more central ponds are more independent of its size, because receives more individuals, the flow it's high, so in a way that there are like a rescue effect. You receive all the time individuals, so the area, I don't know if it could have an effect or not. In peripheral, maybe, probably, yes.
Well, thank you very much, Anna.
Thank you.
It was very great talk. Thank you very much. Also, Pedro include a similar message. Thanks again, Anna. Nice to see again Steven and Luke and Pedro. And Coca, te paso la pelota. Adelante.
Thank you.